Yes. Hello. Thank you so much for having me. I'm delighted to be here, and I'm delighted for all of you who are here on this Friday night. I'm excited to bring you the story about how we were trying to understand sort of the fundamental limits of why cells reprogram and what undergirds plasticity. So reprogramming is, I think, really exciting for a lot of different reasons, especially because provides the potential to think about fulfilling the promise of regenerative medicine by providing cellular therapies, creating cells for disease modeling, and ultimately for therapeutic screening. And so that's sort of the promise of where it's going. But the thing that I think is really scientifically interesting is the fact that we don't really understand this process or exactly all of the limits. And we've seen some lovely talks today thinking about how do we track this process? How do we drive it? How do we quantitatively characterize it? And I'm going to be telling you a little bit today about what we've seen in our specific system where we take fibroblasts, overexpress these transcription factors that make motor neurons, and can then monitor the process in this way. And the thing that got me really excited about this was it's just a fundamental conundrum that's hidden in this cartoon, but revealed when you actually look at the cells in a dish. So clearly you can see here that one of these cells, which these are labeled with HB9-GFP to make motor neurons, one of them looks like a neuron and one does not. One's really trying hard, but it just never really gets there. It's kind of stuck. It's got this arrested development. So this is the reprogramming enigma. We have essentially the same input. It's not a difference in viruses between these two cells, and yet they have these different outcomes. And so the question is why? Why do these cells make different choices? And what I think is, again, interesting about this, I think of this as this problem, at least how we approach this is a developmental synthetic biology where we're trying to build these cellular transitions so we can understand plasticity. And when people look at this problem, I think depending on who you are, this is like a real Rorschach test. Like, what's the limitation? Well, if you're an epigeneticist, it's epigenetics. And if you're a synthetic biologist like I was, I'm like, oh, it's just the genetic programs, build one more sophisticated and it'll work. And neither of those are entirely wrong. But I think that there's a different view that actually allowed us to kind of advance on this problem and think about more of this, not from so much a memory problem, borrowing the analogy from computers like with epigenetics, or a software problem a la synthetic biology, but more as a processing problem, and specifically at the level of DNA. And so I'm going to give you all the talk in hopefully just one slide here. What we found was that there existed this rare population with high processing capacity. And by that, I mean there were cells that could have high transcription rates combined with high proliferation rates. And that population was extremely rare. But if you could expand that population, and as we were able to do, we found that those cells would reprogram very robustly at high rates, and were also marked by signatures of maturity, both at the transcriptional level and functionally. And so look, the quest to understand this was then what are the mechanisms that actually undergird this process, and why is this process rare? How do we promote it? And so I'm going to tell you this is what we found, and I'm going to take you through how we got there, and then where I think it's leading us in terms of how we actually design genetic circuits to facilitate reprogramming and a whole lot of other cellular engineering strategies. So we got started with this work and even thinking about proliferation rate and reprogramming from this paper from 2014, the Guo lab, where they noticed that fast cycling cells were this privileged state in reprogramming. And so if you separate them out based on CFSE, which is a dye that allows you to mark the fast cells based on dilution, so the dim ones are fast, you can see that those are the cells that preferentially reprogram and represent this privileged state. But it was a little bit challenging to figure out if proliferation was actually promoting reprogramming to a fast cycling state, or if it was just, fast cycling cells become fast cycling cells by being fast cycling cells. And so we kind of wanted to ask, well, is fast cycling something that actually promotes reprogramming to post-mitotic fate? And are there other determinants? And we actually did observe that fast cycling promoted reprogramming, but it wasn't deterministic. So it wasn't enough. If you make a fast cycling cell, it doesn't become a neuron right away. And so we asked, what's the other possible determinant? And we turned toward transcription. And so I'm so glad that John talked about how much transcriptional variability there is, because we thought, okay, if proliferation isn't the determinant, could it be the combination of transcription and proliferation that made these cells elite, that made them rare? So we developed an assay. This is a high throughput flow assay where we label our cells with CFSC a day after we add in our factors. And then we allow them to divide for 72 hours after that labeling. And then 4 days post-infection, we feed EU, which allows us then to label our transcripts, the RNA that's being synthesized with the EU incorporation to those transcripts. We then perform a click reaction that allows us to then measure the transcription rate based on the fluorescence. And then we can flow these cells to analyze them for proliferation and transcription. And the first thing we noticed was that our cells that were up here bright, our slow cycling cells had high transcription rates, and our fast cycling cells over here had low transcription rates. And that different genetic perturbations and chemical perturbations moved both the CFSC, so our proliferation, and our transcription rate. And the cells that we call these hypertranscribing, hyperproliferating cells were rare, and their transcription rate was high relative to their place as highly proliferative cells, but it wasn't high in the global scheme of things. And if we hadn't had this determinant of CFSC to observe them, we would entirely have missed them because they're right here in the middle. They kind of look average. They're just your average Joe, but they're punching above their weight for their proliferation status. And so we thought, could these be the cells? And what we noticed is as we expanded this population, we could see so we increased these HHC population with these combinations, and we could see a really huge increase in our reprogramming rate. It's not as compelling when you just see dots on a screen, but when you see the cells in a dish, it's pretty remarkable the difference we were able to achieve. We went from a handful of cells in a dish to really these nice, dense cultures. And ultimately, I won't have time to take you through this, but we did demonstrate that the HHCs are the elite population that preferentially reprograms through a series of somewhat sophisticated experiments that I won't take you through. But what we thought about, thinking more about the mechanism, was how are these cells able then to deal with these high rates of transcription and replication? And what does that mean for us generally wanting to make an elite cell, a highly plastic cell that can do these things? And specifically, we thought if you have a high transcription rate and a high proliferation rate, you are going to be inducing a lot of stress on the DNA via the effects of DNA supercoiling introduced through replication and through transcription. And so if you don't, and you're going to need then enzymes to deal with that. And so we wanted to ask the question, do we actually see the stress that you would expect to observe? And indeed, what we found is that whether it's chromatin bridges or micronuclei, we saw that the introduction of these reprogramming factors induced a lot of genomic stress. Additionally, our reprogramming cocktail, which I'll just refer to as DDRR, a combination of P53, HRAS, and RepSox, a TGF-beta inhibitor, reduced this stress significantly and also limited the formation of these binucleated induced motor neurons, suggesting that it was somehow mitigating this genomic stress. So we wanted to know what are the molecular mechanisms that actually underpin this. What could it be that's labeling these that's allowing these HHCs to perform at these high processing functions? So we went in and performed a single cell RNA sequencing experiment across the reprogramming time. We took fibroblast hyperproliferative cells at day 4, HB9 GFP positive cells at day 8, and then the final induced motor neurons at day 14. Putting them on this landscape, we were able to identify a population of putative HHCs based on their proliferative status. We noticed that they were marked by topoisomerases. Topoisomerases came up as being an interesting hit, and it kind of started to check the boxes for us in terms of thinking about genomic stress. So topoisomerases resolve supercoils that are introduced by transcription and replication, and therefore they'd be important for reducing stress. We knock these down, you lose the HHCs, you lose your reprogramming rate. Additionally, if you treat them with inhibitors instead of shRNAs, similarly, you drop your reprogramming rate. And so these populations of HHCs are supported by these topoisomerases, which gave us some interesting ideas about what it might look like then to think about designing circuits that could actually promote reprogramming. And what I mean by that is we know then that these genomic processes are limiting our transcriptional activity in terms of our ability to induce the gene networks that we want to turn on in reprogramming. So we wanted to ask the question, can we see this at a single genetic locus using a circuit as a test bed system. And I'm going to skip through this because I'm running just a little bit behind. But what I want to tell you is that we took a circuit, a simple inducible circuit, knowing that you could put it into two different orientations, that these two different orientations would induce different levels of genomic entanglement, and then built a model to predict how that genomic entanglement would then manifest as different levels of gene expression. We could see that if in a tandem orientation, a circuit would have very little coupling, whereas our divergent and convergent orientations would start to interfere, suggesting that we could actually, at the level of chromatin, potentially engineer how these elements interact. And again, I'm running out of time, but the thing that I'd like to leave you with is we've got our first really exciting result in that our tandem orientation, where we have this reporter that then is co-expressed with this inducible element. We see that there's this correlation expression, which is exactly what you'd expect based off our model. There shouldn't be really any coupling or any drop. Whereas when we had our convergent orientation, we predict negative feedback through the coupling of these, the physical coupling of these two elements, and you see a drop. And that's exactly what we saw when we just simply flipped around. These are the exact logical exact logically same circuits, and yet their physical coupling is resulting in vastly different expression profiles. We think that's really interesting because this physical coupling should also be controlled by the expression of topoisomerases, potentially meaning our circuits can serve as sensors for these different populations. And if you have high expression of these topoisomerases that we see in HHCs, we'd expect that you'd have high activity, and they'd be able to untangle these circuits. That's what we predict. And we have some preliminary results that show that these populations where you have high expression of both parts of your circuit only exist in the case where you're not inhibiting these topoisomerases with the camptothecin, specifically top1. And we did not observe these results when we inhibited with top2A via doxorubicin. So we're really excited about this. We are enthusiastic to start thinking a lot more about how do we physically engineer circuits. Most of the work in synthetic biology is focused on the logical functions and the organization or dimerization. But I think the physical realization is something that is, it's the right time for us to start looking at this, and especially using the new genomics tools, including micro-C, to look at the structures. So with that, I would just like to conclude with this is all in support of our goal of actually building circuits that can support regenerative medicine. I'm grateful, especially to my lab, who has endured a very challenging year. I started in July 2019, and my students started in 2020 with me in January, and we've grown very rapidly. We're still recruiting, and I'd be glad if you could, for those who are interested in joining us. Also, this story started in the Acheta lab at USC, where I was a postdoc, and I'm thankful to Justin and especially Kim Wonder, who's a graduate student on this project. With that, thank you for your time and attention, and I'm glad to take questions if we have time.
Thank you, Katie, for a wonderful talk. We've got time for a little off, but we can do a couple of questions. So King Sun asks, or says, super cool work. I want to ask, how precise is the reprogramming? And do hyperproliferative cells also give a higher risk to form cancer?
Yes. So how precise is the process? I mean, I think we're still only reprogramming. If you think about precision in terms of like how many cells can we reprogram in our dish, we can make about 33 or 1/3 of the cells be HHCs, and those seem to reprogram, highly efficiently. But I'd say it's still not that well controlled. And I think we can do better. That's the aim of a lot of what we're thinking about with these gene circuits, to control the reprogramming process. But we feel like the circuits themselves have to be well controlled and well behaved within the genetic environment. And that's not something in 3D we think is actually achievable yet. But we're mapping that out. And in terms of cancer, yes, absolutely. Like I think regeneration, plasticity, cancer development, they're all balanced on an edge of a knife. Anything that will allow you to transition in one direction is essentially the opportunity to transition into something that I guess to de-differentiate. And I think that always makes us wonder if you're headed towards cancer. So to be able to control those processes for regeneration, I think is inherently balancing the risk with cancer. At least that's my personal thought on it.
Cool. We'll do one more question. So, Geetika Arakatia asks, cool talk. Are the elite cells always elite? Or did you happen to catch them at the right time? Basically, does every fibroblast have the potential to be elite at some level?
Ooh, I love this question. We ask this question all the time. Like, would you, if you rolled the dice and seeded the cells a different way, would the cell save cells reprogram, or is there some sort of ergodic behavior in which the cells would come back, and it just depends on when you would hit them. But the distribution itself is kind of fixed, but any given cell's potential is not. I tend to think that's the case, but I don't know if we have any evidence to support that. And I think that's even thinking about the experiment you would do to test that is, I think, a little bit complicated. With proliferation, it's obviously true. If you take the fast cycling cells and then seed them, they'll recapitulate the distribution that they came from. So in reprogramming, we have an arrow in time in which there's irreversibility. So it's hard to ask that question. But certainly with proliferation, it's true, if that's our only determinant.
Yeah, such a fascinating question. I don't know. I always kind of wonder about it. Anyway, thank you again, Katie, for a wonderful
Thank you.
talk.